All right, well, welcome everybody. I'm Professor Nate Snydecki in Mechanical Engineering, and this is our first seminar for Mechanical Engineering 520, which is our department seminar. We have a really great speaker for our first seminar of the quarter. Ayo Olanrewaju, I apologize, I tried to do it the right way. He is an acting assistant professor in our department, new this year, so he has that squeaky clean look on him for that. Ayo has been an engineer all his life. He did his bachelor's in electrical engineering at the University of Alberta, his master's in biomedical engineering in 2011, also at the same institute. He then went over to McGill University, where he got his PhD in biology and biomedical engineering with David Junker. And then he came to the UW, and I believe it was 2018, as a postdoc in mechanical engineering with Professor Jonathan Posner here. Ayo has done some amazing work, both as a postdoc as well as his previous stints up in Canada. Looking over his CV, you'll see a lot of great accomplishments, but the two of them that I thought were really cool was Ayo is a great speaker, and he's been recognized for that. He got first place for a competition, a Shark Tank competition at one of my favorite conferences, Microtas, in 2017. He also got first place for an elevator pitch at another fantastic conference, the American Society for Cell Biology in 2019. So I don't know how he does it, but what he says impresses people. And not only what he says, but also what he writes. So he's also been very successful at proposals. He just recently got an award from the UW and Fred Hutch for AIDS research. They're a new investigator award for 2 years, so congratulations. That's fantastic news. We're all really excited about that. And I'll let Ayo say more about what he's been doing and what he will be doing here at UW. But it's a great pleasure to have you, and it's great to have you as our first speaker of the seminar. Just as a reminder, this is recorded, and this is mostly for those who can't make it. If you do have questions for Ayo, we will have a Q&A afterwards for those questions. So I'd ask that you hold your questions until then. You're welcome to use the chat, or we can have you raise your hand and then you can ask your question as well. Are there any other ground rules, Ayo, that you'd like us to follow?
I know that sounds good. I'm happy to answer questions via the chat or raise hand. I know everybody's in Zoom a lot, so if you have questions during, I'm happy to pause and answer. And Nate, if you don't mind helping me look at the raise hand feature.
I will. It's always tricky because I know when you're presenting, you don't see all the features. So I'll do my best to be an assistant to you.
Awesome, thank you.
All right, well, I will go blank and I'll let you take the show.
Great. Thank you so much for such a kind introduction, Nate. And thank you everybody for being here. I know it's 3:30 on a Tuesday, so I imagine energy levels are a little low. I'll try to give an engaging talk. But yeah, thank you all for being here. And I'm really excited to kick off the departmental seminar series for ME. And today I'm going to be telling you about some of the work that we're doing here at UW that's focused on developing a rapid and accessible test, excuse me, for measuring antiretroviral drug levels to improve HIV treatment and prevention. And so to start, I really wanted to sort of set the stage for you. So imagine that you're a physician, right? And so imagine that you're a physician and your patient comes in and they're coming in for a regular checkup and they don't seem to be getting better. You have their next round of prescriptions, but yeah, they just don't seem to be getting better and you're not quite sure why. And there's a few things you're wondering. You're wondering, A, are the medications working? You're wondering, B, is there enough in their system? Do I need to up their dose? C, you're probably wondering, are they developing some sort of drug resistance to this? And trying to answer those questions can be challenging, and it can be additionally challenging if you're in a low or middle income country where you don't have enough resources to run all the lab tests you want to run. It can also be challenging if you see your patients infrequently and you don't want this to be something that gets progressively worse. So you want more tools, you want more diagnostics that allow you to really personalize the treatment that you're giving to this patient, reduce harm, and avoid adverse side effects. So this is something that extends beyond HIV, but today I want to talk to you about it in the context of HIV. And so the bulk of my presentation, or the outline of my presentation, is I'll start off by telling you a little bit about HIV and antiretroviral drugs. I know that we all, come from several different backgrounds in terms of areas of expertise. So I'll give you some information about HIV, about what the drugs that we use are right now and how they work. And then I'll tell you about an assay that we're calling REstrict. It's an acronym. I'll explain the acronym, but I'll tell you about how that assay works and how it relies on the mechanism of action of the drugs that we're interested in. I'll tell you about how we do this assay with blood samples. I don't know how many of you have worked with blood or tried to diagnose something in blood, but it can be messy. And so I'll talk to you about some of the things that we do to get this to work well in blood. And then finally, I'll talk to you about ongoing work and next steps and some of the things I'm really looking forward to us doing. So I wanted to start with a bit of a pop quiz. And I wanted to gauge, and feel free to type in the chat, how many people roughly do you think live with HIV across the world? And we can answer sort of Price is Right rules. The closest to the answer without going over will win today's Zoom prize. So how many people around the world are currently living with HIV? This is as of 2018 estimates. Just to get a sense of the scale of this problem. 200,000, 150K, I would say go higher, much higher. 1 million, I'd say go higher. How many people living with HIV? Ooh, I need to count the zeros. 35 million, very close, 35 million. So the answer, it's a little higher than that. It's 38 million as of 2018. So about 38 million people living with HIV. And it's really interesting to think about the distribution as well. And so the distribution is predominantly in Africa, in sub-Saharan Africa, in the Americas, Southeast Asia. It touches every continent. I guess Antarctica is not in here, but it touches pretty much the whole world. And what's interesting to think about is this distribution is not a result of people being differentially susceptible to HIV, but it's often that HIV affects and spreads amongst the most vulnerable in society. So the correlations between income inequality, between education, between access to quality healthcare and HIV rates are staggering. And so a lot of this distribution is that this is a disease that's endemic, especially in places that are lower and middle income. And even in the US, areas with high rates of poverty tend to have high rates of HIV because of education, lack of education around how it spreads, lack of access to quality healthcare and prevention methods. So it's a major, major health problem around the world. And what does HIV do exactly? So it's a virus that attacks your immune system, and over the course of years, damages your CD4 immune cells, which protect you from other infections. And so what happens is you get acquired immunodeficiency syndrome, which is basically a situation where you've lost a core component of your defense system. And people actually don't die from AIDS, but they die from opportunistic infections that occur when you have AIDS. So one of the most common causes of death for people that have AIDS is tuberculosis. So a lot of infections that people don't normally get, they will get when they have AIDS because they don't have the immune systems that would protect them. So a lot of bad news so far. Let's switch to some good news. So some good news is that we have antiretroviral therapy that can lead to viral suppression. So what I mean by viral suppression is you take a combination of medicines that reduce how much HIV virus is in your system to an undetectable level. Now, this is crucial. It's not a cure, but it's a way to reduce how much HIV virus is in your system to a level where you're protected, your immune system recovers, it doesn't progress to AIDS. And interestingly, actually, if you have an undetectable level, it's not transmittable. So there's been a lot of really interesting work that's been done over the past decade that's shown that if you have an undetectable viral load, you can't transmit it to somebody else, which is fantastic. And this is really, really interesting work that people are doing. And beyond that, there's also work done in PrEP, pre-exposure prophylaxis, where if somebody's at risk of HIV, you can take HIV medication preemptively, regularly, and even if they're HIV exposure events, you do not get HIV because you have medications in place to fight the virus before you're exposed to it. I don't know how many people in the Seattle area have seen these chill pill posters. I used to live in Capitol Hill and a lot of them were around, but there's a lot of PrEP rollout happening and it's happening locally here too, which is great. There's a lot of treatment, there's a lot of prevention, and that's going a long way to reducing HIV rates. I wanted to talk about the drugs themselves because this is an interdisciplinary area and understanding how these drugs work will be very, very crucial in understanding what kind of tests we make for them. So the first ever approved HIV drug was a drug called azidotimidine, or AZT. It was approved in 1987. And the drug is part of this family called nucleoside analogs. And I'm going to play a game of Where's Waldo. I don't know if you can see my screen well enough, but can you tell the difference between AZT and timidine? So timidine is a base, it's a nucleotide that's used to make DNA, and AZT is a drug and it's a nucleotide analog. So can you distinguish between the two? What's the difference? Where's Waldo here? So I know you probably can't control, yeah. So methyl group, yes, but I think actually this might just be the chemical drawing didn't show this fully. There's something else that's a Where's Waldo difference. I think this is also a methyl group, but they just didn't spell it out. There's another difference between the two. Yeah, the nitrogen. Yeah, so the trinitrogen group. And this is important because what it means for the drug is when you have normal DNA synthesis with a nucleotide, you can incorporate lots of nucleotides and form a chain. But when you have a drug like AZT that has that kind of substitution, you can't form a chain. That trinitrogen group will not react. And what you get is a stop. You get DNA chain termination. And so drugs like AZT, nucleotide analogs, are really designed, I use nucleoside and nucleotide analog interchangeably. There's a technical difference, but it doesn't matter too much for our case. But really, they terminate DNA chain formation. And that means that HIV cannot turn your cell into HIV virus factories. And that's one way you can stop the infection. There's other drugs that are being used, but really drugs in this class are still the dominant drug. So the new drug that's used right now is a drug called Tenofovir. It's also a nucleotide analog. It's used in over 90% of treatment regimens and all the approved prevention regimens right now. Again, tenofovir diphosphate looks a lot like DATP. So this looks like an A. And I can't tell if the screen is clear enough, but we can do a similar Where's Waldo for this one if you're interested. What's the difference between tenofovir diphosphate here on the left and DATP on the right? I can do a spoiler. So it's really, here you've got a methyl group and here you've got a sugar ring. And again, it's a similar thing where you've got a reactive group and a non-reactive group. So that's how the drugs work. And what impact have they had on global health? So the impact that they've had is since 2000, there's been this massive rollout of antiretroviral therapy. In fact, we're now at about 25 million people receiving antiretroviral therapy, which is remarkable. It's really, really impressive. And what that has meant in terms of prevention of new HIV infections is it's been a dramatic drop in the number of new HIV infections, dramatic drop in the number of HIV deaths every year, which is fantastic. We're making a lot of progress. We're not there yet. So the UN set these 2020, 1990 targets. And what those targets were was they wanted 90% of people who had HIV to know their status, to have received HIV diagnostic tests. They wanted 90% of those people to be receiving treatment. And then wanted another 90% of that 90 to be virally suppressed. And you can see that there's still some gaps. There's gaps in people that we still haven't covered yet with testing and treatment. There's also gaps in people receiving treatment where the treatment isn't working. And so the question that leads to is why is treatment failure occurring? Why are there people on treatment where they're still getting sick? They're still progressing to AIDS. And there are three key reasons. There's a lot of societal reasons, but biologically there are three reasons. So one of them is drug toxicity. And so the idea here is people metabolize drugs differently. You and I probably have different metabolisms in terms of how we metabolize drugs. I guess one example would be what's the famous one? Is it not celery? There's something that tastes like soap, cilantro. Yeah, so I guess that's a famous example of some people really don't like cilantro. It tastes like soap. I feel like that's probably driven some 23andMe tests that people have run where, so we metabolize things differently. And so for some people, what would be a normal dose would be a toxic dose. Now, the catch is drug toxicity and adverse effects can lead people to take their medications less. Now, taking your medications less, unfortunately, then provides ideal conditions for drug resistance to occur. So if you have a low level of HIV, of HIV medication, it could be low enough such that the virus can survive and actually develop drug resistance and start to thrive, even when you bring in higher drug levels. And so these reasons can sometimes interchange and interplay. Poor adherence is not always caused by drug toxicity, and drug resistance is often caused by poor adherence, but not always. But the key thing here is to figure out why treatment failure has occurred. One thing that would be very, very helpful is to know, does this person have a high enough level of drug in their system? Is it too low, and is there a risk of resistance? And so studies that are done right now, clinical trials and pharmacokinetic trials, they basically take somebody's blood, measure how much drug is present intracellularly, how much active drug do you have? And people have been able to make inferences. Like, for example, if you take above 4 doses a week, your risk of HIV incidence, if you're on PrEP, essentially drops to 0. If you have less than 2 doses a week, it's almost like you're not really taking any of your medication. And if you're in between, it's a low risk, but it's still higher than if you're taking all your medication. And so we've done studies like this where we can actually identify roughly, for most people, what's a concentration of drug that's protective. Another thing that we've been able to identify, though, is that there are demographic factors that influence how we metabolize HIV medication. So here's a study that came out earlier this year that looked at what drug levels were in men and women, in males and females, adjusted for age, race, body mass index, hematocrit, which is a red blood cell count, and a variety of other factors. And on the right here is the study looked at what drugs were used in combination with the nucleotide analogs. There's a lot of different classes, but what you'll find is actually if you use different drugs in combination, so PI here is a protease inhibitor that targets a different kind of enzyme, you can get very different drug levels. And this can play into things like adverse effects and toxicity that can lead to people not taking their medications. So having an understanding of what people's drug levels can be useful in tailoring their treatment. So we know this information is useful, but all of these studies that I've showed you so far around drug levels were all done with complex and expensive procedures. So to really get a sense of drug levels, you have to spin the blood to separate out red blood cells and white blood cells. You do liquid chromatography, you do mass spectrometry. And it's very accurate, you get really quantitative results, but it takes about 4 weeks because there's only 3 labs around the world that can do this particular test. It costs about $200 a test. And that's prohibitive because where we really need HIV drug level tests are in near patient settings. So for example, the Gay City Clinic in Seattle, or the UW has a Center for Integrated Health of Women, Adolescents, and Children in Kenya. And what you want to be able to do is when somebody comes in for a refill of their prescriptions, you want to be able to measure what their medication levels are right there and then and adjust as needed, rather than sending it out for a month and then waiting to see what happens. And so this is where I get into what we do here at UW, the REstrict assay. And so when I came to UW 2 years ago to work with Jonathan in the mechanical engineering department, I was working on a project with Dr. Paul Drain, who's a clinician and an epidemiologist, and he goes and does validation studies of new point of care tests in a variety of settings. So they do work in South Africa, they do work in Kenya, and really what they're trying to figure out is what role do point of care and near patient diagnostics have in improving the kind of care that people get. And where we settled very quickly was you said, can we use the ability of these drugs? Can we exploit the DNA chain termination property of nucleotide analogs to measure HIV drug concentrations? That was the key idea that we had. And so we came up with what we call the REverSe TRanscrIptase Chain Termination assay. And the key idea here is that we provide a tube that has everything HIV needs to make more DNA. So we give it a DNA template, we give it a primer to tell it where to start making DNA, we give it nucleotides that are DNA building blocks, we give it the drug of interest sometimes if we're interested. And then we put in HIV reverse transcriptase enzyme. That's really the enzyme that does the heavy lifting of making DNA. And finally, there's a dye that tells us if DNA was made. Now we control everything that's going on in this reaction except for the drug level in a patient's blood. The idea is that by setting up this reaction, if a patient has high or low drug levels, we can distinguish. So this next slide is sort of a schematic equivalent of that. So on the bottom here, if somebody has a lot of drug, chain termination occurs early and you don't get much signal. Conversely, if they have not much drug in their system, you form a lot of DNA, your dye binds, and you get a high signal. And if you're somewhere in between, you're somewhere in between. So really, this is sort of the key idea of this assay. Lots of drug, no signal, not much drug, high signal. And the idea is that we can distinguish these different drug levels with this kind of assay. Now, we don't want to just do this in an intuitive sense. We want to be able to do it deterministically and systematically too. And so it actually made us think that maybe we can represent this probabilistically. And so the way that we thought about this is, say you have black ping pong balls and yellow ping pong balls. The black ping pong balls here represent the drug. The yellow ones represent your nucleotides. And so we put them in a little bucket, shake, 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 you put your hand in it. And so if I asked you, what is the probability that I pick the drug out of this bucket? Many of us can calculate it. It'll just be 1 in 9, right? Now, what about a scenario where somebody had 3 ping pong balls, right, that were black? What's the probability that you pick a black one? 1 in 3. So you can start to think about how we can probabilistically represent this. Now, say we want to vary our assay around and we want to be able to minimize this probability. Say I want it to be much less likely that I pick a black ping pong ball out of this bucket. What are some things I could do? What are some knobs I can tweak? What are some things? Because I control everything except the number of black ping pong balls. So what would I do? Say I want to make this a very unlikely event. What are some things I can do? So we thought about this. One of the things, this probably, I guess, has occurred to some of you as we're thinking about this, is just get a bigger bucket and fill it with a lot more yellow ping pong balls. So that's one of the things you could do, right? So if you put a lot more here, your probability goes down. You can imagine a scenario where you can also go the other direction, where you make it much more likely. And so what we were able to do was we were able to design this assay and represent it with equations. And so this is work I actually did with a grad student in our lab, Ben Sullivan, where we said, how do we represent this assay probabilistically and systematically so that it's repeatable? This is the only equation slide I have, so no worries if you're not about the equations on the slide. Really what this equation is saying is that the probability of picking DNTP every single time, so the probability of forming the full-length DNA template, depends on how much DNTP you have divided by how much DNTP plus how much nucleotide analog. There's a factor here, this affinity factor that we're calling the relative affinity factor of your enzyme for your drug. And really the way you can think about that intuitively is you're reaching into this bucket. Now imagine that the black ping pong balls are a little bit softer, so you can actually tell, so you're not completely blind to it. So it's possible that the drugs are not exactly like your naturally occurring nucleotides, and your HIV enzyme can actually distinguish a little bit. So we can represent this assay probabilistically, and that allows us to also have some theory that guides our design. So what do we actually do in the lab? So when we're optimizing the assays, we prepare a master mix that contains everything. We pipette into a well plate because we're trying to test as many different conditions as possible. We incubate for 30 minutes at body temperature, and then we read out results. And here's what our results look like. So it follows the expected trend where we have at high drug concentrations, a low signal, and at low drug concentrations, a high signal. I've overlaid here what the clinical range is, around how many doses a week you have. So you really want to be distinguishing somebody that's up on this end from somebody that's down on that end. And the other thing that I've labeled on this slide is what we call the 50% inhibition concentration. What that means is how much drug do you need to get a 50% signal on the assay. And this is a parameter that people use in enzyme inhibition assays as a way to see, how far left do I need to go? How far right do I need to go? So it's sort of a variable that we use. Next, we said, well, let's optimize this assay. How far can we move it? Can we move it left? Can we move it right? So this experiment really was the equivalent of how many yellow ping pong balls do you have? Intuitively, what that means is that very low DNTP concentrations, you're very likely to detect the presence of low concentrations of drug. Meanwhile, at high DNTP concentrations, you can only detect high concentrations of drug. If we map this according to this 50% inhibition concentration, things are a little bit clearer. The relationships are slightly clearer. So what this tells us is, as we increase DNTP concentration, our 50% inhibition concentration also increases. And then you can change other variables. So say you change your DNA template length. As you increase your DNA template length, your 50% inhibition concentration decreases. And there are intuitive explanations for this. So one way of thinking about this is to say, if I have a really long DNA template, it's almost like I'm giving myself more opportunities to dip into the bucket, right? And so say I have a bucket and I go in 1 time, 2 times, 3 times, 4 times. The more times I go in, the more likely I am to detect even small amounts of what I'm interested in. So that's what this means. The longer the template, the more likely you are to detect even small concentrations of the drug that you're looking for. And so being able to do this both experimentally and theoretically really gave us a lot of confidence that we had this assay at a point where we could actually start to throw some real samples at it and say, can we work with blood? So moving into blood. Where do we expect to find these drugs? We expect to find them in the white blood cells, in the immune cells that are targeted. But serendipitously, people also found that the drugs actually also build up in red blood cells. And what's the advantage of having these in red blood cells? One, red blood cells are abundant. There's a ton of them, much more, about 1,000 times more than white blood cells, which means that you can find more drug in your system. But the other thing is red blood cells are really, really wimpy. And so what that means is if I take blood or red blood cells, put them in water, they swell up due to the osmotic pressure change, and they burst, and they release the drug that you're interested in. And so this is work that I did with Dr. Jane Zhang in our lab. And what our workflow then looks like is we collect blood, we dilute the blood to release the drug of interest, we add that diluted blood to our assay, we incubate it and read it out as we did for everything else. This is a process where everything from having your sample ready to running your assay, we can do the whole thing in about an hour. And when we do this in blood, so we take tenofovir diphosphate, the drug we're interested in, we spike it into blood, and then we do this at several different concentrations. And we still get that characteristic sigmoidal shape that we're interested in. Now, some of you might notice that there's more noise, and this is in part because there's a lot of other stuff in blood. There's antibodies, there's hemoglobin, but we're very, very encouraged to see that you still get that same shape that we expect, where high concentrations, not much signal, low concentrations, high signal. And if we zoom in a little bit in the region that we're interested in, we see that we can actually distinguish concentrations that correspond to 1 dose a week from 7 doses a week when we do this with spiked blood samples. And so spiked blood samples are one thing, but can we do this with actual patient samples? How close can we get to real world conditions? So we're very grateful that we have the Center for AIDS Research here at the Madison Clinic at the Harborview Medical Center. And so they have research coordinators. So Lindsay and Mark Jason recruited PrEP clients for us, people who are taking PrEP. They did blood draws. And then I would just show up back when we could go places. I would show up, take a little cooler with blood, go back to our lab and test them. And what we found in this little pilot evaluation, just to see, is this feasible? Can we do this? Is first of all, we compared fluorescence that we got from the restrict assay on the y-axis with measurements from mass spectrometry, which is the gold standard. And what we found was that there was a correlation. Yes, there was a lot of person-to-person spread, but there was a correlation between the restrict fluorescence and the mass spectrometry measurements. And then what we also looked at was we looked at, can we tell with restrict, can we find a cutoff that distinguished between people that had drug concentrations above 4 doses a week and drug concentrations less than 4 doses a week. And we were also able to have a threshold where we could distinguish. And it's a very small sample size. I think we had 18 people in this initial study, but we're excited to continue doing this and continue to do more tests. So I want to tell you about some of the things that we want to do next. I think first off the bat is we want to do a larger clinical study where we look at more patients. So this image on the left is from a landmark paper from 2012 that was one of the first to actually demonstrate that PrEP was effective. And one of the things that they found was you can see drug levels corresponding to 2 doses a week are different from 4 doses a week or different from 7 doses a week. This is all done with mass spec, but you can see that there's a lot of person-to-person spread, but still the overall drug levels are different. And then you can see that people who, while they were on PrEP, developed HIV, have drug levels that are closer to 2 doses a week. Meanwhile, people who don't, the controls who didn't develop HIV, had higher drug levels. But again, there's a lot of spread. And so one of the things we want to do to really show that Restrict can do similarly is to recruit patients. So we're going to be working with Dr. Paul Drain, Dr. Alex Greninger, who's at UW Lab Medicine in virology, and then Dr. Pete Anderson, who's actually one of the authors of this paper and one of the leaders of a lot of the mass spec work for measuring antiretroviral drug levels. And so our plan is to recruit patients. We submitted a grant to the NIH in May and we got our score back and it's looking promising. So hopefully we'll get some funding to scale this work up and continue developing it. But we don't just want to do future work and test more samples. In the meantime, while we wait to do that, we're also working to make this assay better. So we know that there are areas that we need to improve. One of them is, we actually have 4 students in the lab right now that are working with me on making the Restrict assay better. So Alicia is actually optimizing our theoretical model and trying to package it in a way that not only automates several different software tools that we need, but also in a way that we could share with other people and allow them to actually design their own assays without having access to what we do. Jason's designing templates that can detect even lower concentrations of drug. Alicia is doing multiplexed assays where we're looking not just at tenofovir diphosphate, but other drugs of interest. And then finally, Jonas is looking at using a low-cost reader that will allow us to do this, not just with a plate reader, but with something that costs much less and is a much smaller form factor. The longer term goal for the project though, a lot of what I've said, is very lab-based and it's simple, but it's still lab-based. But where we really want to go is how do we do this at the point of care? How do we do this without the instrumentation? How do we do this in a way where somebody could run this directly in the field? So for me, one of the areas I'm really excited about is to bring in some of the work I did in the past where we looked at microchips that can be used for complex bioassays. And the idea is to have a microchip that's, size of a credit card that automates all the steps you need, that automates the dilution, automates the mixing of your master mix, automates the incubation step, and automates the final addition of dye. And really the goal would be to do this in a simple form factor, whether it's in a microchip or whether dried in a little tube, trying to figure out how we can do this in a way where we can actually deliver it right where it's needed. And on that note, I also wanted to mention that our lab has other projects that are going on. So we've got a point of care viral load monitoring assay. We've got a COVID home testing assay that we're doing. There are also people working on tactile sensors for artificial skin. We're hiring at all levels, undergrad, master's, PhD, postdoc. Come and work with us. It's a fun group to be in. And if you're interested, you can go to the Posner Lab website and there's a contact us button. You can also reach out to me and I'm happy to chat and link you up. There's more work than people. And so we're pretty excited to get more people on board. And I just wanted to conclude and kind of summarize what I've told you so far today. So hopefully you'll remember that nucleotide analogs cause DNA chain termination. And that's a neat way to set up this Restrict assay where we can correlate drug levels we can infer drug levels from DNA synthesis reactions. We can optimize this assay with a theoretical model where these dashed lines are actually our theoretical model and the open symbols are experimental data. And finally, this allows us to distinguish different drug levels within patient samples. And really our goal with this is finally to get to a point where we can meet this doctor's need, right? Where we can get to a point where a doctor says, well, is this working? And we can say, actually, in a matter of minutes, yes, it's working. There are a lot of people to thank because I'm not the only one that's worked on this. I'm going to thank Jonathan Posner for all his support and mentorship. And we've got a really good group of people here who have been very helpful. Dr. Paul Drain has been a great clinical mentor to have because I think this kind of work really benefits from bringing the engineering and the medicine together. And those two inputs have really been super helpful in how I think about this work and what we want to do next. We have the Center for AIDS Research who support us financially, but also in kind by providing access to patient samples. We've got collaborators. We're not the only ones doing this kind of work at UW. So in bioengineering, Dr. Barry Lutz's lab is also doing a lot of diagnostics and point of care diagnostics. And it's been really great to see areas where there's synergy, right? Where they're looking at how much virus is present, and we're looking, for example, at how much drug is present. So it's been fun to work with them, learn from them, and work together. And so there's other people in Boston, lots of collaborators, and lots of funding agencies to mention as well. But with that, I'm happy to take your questions. Here's my email address. If I realize I'm speaking really quickly, here's my email address if you have any thoughts, questions, comments, and I'm more than happy to take your questions at this point.
Thank you very much. I'm going to give you a round of applause for everybody. Thanks. Any questions from our audience? Again, you can use the chat feature if you like, or raise your hand, or just unmute yourself too. I'll start off while people are maybe formulating their thoughts, Ayo. With the restrict assay, it reminds me a lot of like when you do DNA sequencing and you use like the Sanger method. Are they the same, are very similar to each other in that sense? I mean, because one, you're using this nucleotide analog, but with like the Sanger method, you're using like a dideoxy, nucleic acid. So it also terminates the chain too. Is that, would you consider them very similar?
That's a good question. I think in terms of, practically the chain termination aspect, I think they are similar. I feel like I need to brush up on the exact mechanics of the Sanger sequencing. I think they're similar in that sense. I think where they're different is that restrict is less granular. So in that case, you're kind of looking, you want the ability to be able to tell, one, sort of base pair by base pair. Like you want as much resolution as you can get. I think for us, we're very interested just in a broad, is there a lot, is there none? In fact, if we could help it, we would just say what fraction of our DNA product is full length product and what fraction is completely terminated. So the idea is similar, but the reason why we don't want much granularity is we want it to be as simple and as user-friendly as possible. And so we want it to be something where, basically it needs minimal infrastructure, minimal processing. You just want to be able to get some sort of rough idea. So I think it's an interesting link that you make with the termination. I think one of the things that I like about the way that we've approached this work is there's a lot that's been done in DNA analysis that I think we can bring in and say, how can we apply this? How can we make it something that's translated? So I think that portion is similar, but I think for us, the goal is just simplify it as much as we can.
Thanks.
Yeah, thank you.
There's a question from Angela. Angela, do you want to go ahead and unmute yourself and ask the question?
Yeah, sure. I was just kind of curious if the ultimate goal is to develop the technology to a level where you can do clinical trials and kind of spin a startup out of the lab, or is the idea to just kind of like get a proof of concept working and hope that someone else picks up the reins?
That is a great question. Thank you, Angela. So we've had conversations about this. I can tell you that we've submitted, I think, 2 patents now around this work. And over the summer, one of the students that we had, Alicia, was supported by a Commotion Mary Gates Innovation Scholarship, which is very much targeted around doing work to fill the gap between what's done in the lab and what you could spin out. We also had, I didn't know that this program existed, but it's a similar Commotion business internship for the summer where we had a Master's of Engineering student from bioengineering who looked at the business case for the assay. So I think my personal goal, and I think the goal of a lot of us involved in the project, is to make sure something like this can go out because there is a big need for it. So we want it to be able to go out. Now the question of what form that takes, I don't know that we've answered completely yet. I think one tricky thing with HIV and global health is that it's not a huge moneymaker, right? It's primarily targeted at low and middle income countries. And so by itself, it's not gonna be the thing that makes somebody $100, $200 million. But at the same time, that's not really the goal. The goal is sort of the mission-driven thing where there's a big need and potential high impact. And so one of the ways that we're thinking about it is, who would pay for this kind of test? Often the answer is governments and also NGOs that already pay for the drugs that people are supposed to be using. So we also, as part of this commotion process, have had conversations with people at NGOs, conversations with people in companies active in the global health space. I feel like given the experience we had over the summer with the commotion intern, I'm pretty confident we'll probably be asking for another one for next term. So I think we're actively thinking about this. I don't know whether it will be a startup or a licensing type situation, but we're very keen to move it out. I think a key bottleneck to really demonstrate to anybody interested that this is useful is to do a larger clinical validation study. So I think once we've done that, then I think there's going to be a lot more conversation around, oh, do we want to license this? Do we want to start it up? But yeah, we're definitely thinking about that. And thank you for your question.
Yeah, that makes sense. Now you just need the Gates Foundation to back you.
Yes, yes. Well, we're in Seattle, which is a good place to be for global health. So.
Other questions? I
know the Zoom, oh, what's the cost target for this tech? Thanks, Tyler. So cost is actually also another tricky thing with this. We're talking about wanting to use this in a low and middle income country. But that also, there's a lot of variation in what low and middle income country means. So I grew up in Nigeria, which by definition is a low and middle income country. And you can drive for 2 hours in the same city in Lagos, which is the biggest city in Nigeria, and go from very fancy to very poor. And so there's a lot of distribution. And so the question is, where do you need this? Where's the test needed? And so one of the places we know that tests like these are needed are field clinics where you see people, right? So you know that you need this in field clinics, but you also need this in hospitals, for example, where they might have some sort of lab facility, but they want to be able to do a test very quickly. In terms of cost per test and what people can pay and what's affordable, I think the target that I've heard from collaborators and people that we've worked with. So we've talked to somebody at the Foundation for Innovative New Diagnostics in Switzerland, where basically this is an NGO that goes on the ground, finds need, figures out how much people can pay. The target cost cannot exceed between $5 and $10 for this to be a practical test. If it starts to go beyond $5 or $10, then just the NGOs that would pay for it can really not afford it. We've tried to make the case that, well, look, if you're paying for antiretroviral therapy, and we know that maybe 25% of people don't actually take their antiretroviral therapy, and so that leads to treatment failure, and then you have to pay for more medication. Maybe one way of thinking about the cost is as a way to save medication costs, but I think to answer your question, there's not a firm answer, but it can't really exceed $5 to $10. And then for reference, tests that are currently being used to monitor HIV viral load, which is a different thing than what we're doing, but this is just another HIV prognostic test. Those sometimes cost between $15 and $20, and sometimes there's an instrument cost associated. So we're really targeting $5 to $10. We don't want an instrument. We want it to be cheap. We want it to be able to be deployed in as many places as possible. A flip side to the cost thing, though, is we've done focus groups. So Paul Drain's group has done focus groups at Harborview among people receiving PrEP. And people receiving PrEP have actually said, it would be really great for me if I could just in my house see how much medication I have to know if I'm protected. And so there's a different market there where there's a low and middle income country market, but there's also people who receive PrEP who are like, hey, look, it'd be great for me to know how much medication is in my system. And then there, your cost proposition is different. So maybe you could offer a standalone $10, $20 test that you go to the pharmacy, you buy. So somewhere in that range.
Ayo, can you talk a little bit about some of the challenges in deployment? I think you kind of mentioned it, but you had sort of this product vision, sort of lab on a chip, all-in-one system. Maybe can you describe a little bit about sort of where it is now in terms of like, you got a pipette, you got to use use some small, supplies, whatever, versus kind of what can be potentially
Absolutely.
automated or at least semi-automated?
I like that question a lot because I think it's something we're thinking about, and it's something that as we're writing grants, we're sort of talking about a lot. So what we do right now is, you take blood, usually a vial of blood, you dilute it, and then you add it to a tube, and then you put it in a plate and put it in a reader. Now, a lot of these processes are designed primarily for throughput because we're just trying to say what are the best conditions, or we're going to test 5, 6, 7 samples today. What's the most efficient way to test all those 5, 6, 7 samples? But if you're in the field and you want to do this directly at the point of care, what you want to imagine is something closer to those like a blood glucose test, right? Where you do a finger prick, put it on a little strip or something along those lines, and it's all self-contained. You get some sort of readout. So that's where we want to go. Now, some of these things are really, really easy to replace. So for example, the incubate and readout, we don't need to do a plate. We don't need a big reader. All we need is body temperature and some sort of little tube. So a lot of that, this we can change. Some people have actually published papers where they've done 37-degree incubation, like with armpit warming, but I don't know how reproducible that is. So we're not going to go down that route. But so this part is really easy to replace. The dilution master mix preparation, the way people normally address this is they just dry down all your reagents. So your DNA, your nucleotides, a lot of what you need is actually very stable. So when we buy DNA, it actually just gets shipped from Iowa and it's like in front of our lab for a few days. Like it's super stable in that condition. So you can dry a lot of that down. So you can imagine a scenario where, I'm looking for the chip that I showed, where maybe you don't necessarily even need a full-blown chip. Maybe the thing you need the chip for is timing. So there's a timing component where you need to do this for 30 minutes and then you add the dye. And so the timing component can just be, we've got a chip where you add your sample and the shape of your channel just dictates how long it takes for your sample to flow. And while it's flowing, that's your incubation time. And what we envision with the microfluidic solution is this is sort of a hands-on thing where it's just the shape of your channel and the position of your channels determines what liquid goes where. And so the deployment challenge technologically seems straightforward. I think in terms of getting it to the field and getting it in a cost and a form factor that's like acceptable, that's another thing. So I think I'm really trying to go now to like conferences and talk to people who are more on the clinical side to say, what does your workflow look like? Or what are things that people have developed commercially that have actually made it to the field? What are some of the constraints? What are some of the materials that you can use? What's user-friendly? And so this is another place where it's nice to be in Seattle because PATH, so there's the, I don't know what, so it's another NGO that's very active in the global health space. And they do a lot of user experience studies, for example, where they'll say, here's something we know will be useful. So our approach here would be, once we have a technology that seems to work as a proof of concept, we want to very early on engage people from industry, engage actual clinicians on the ground, and find out what's their wish list? What's something where absolutely they must have this? A lot of that might happen outside the lab, but I think just to kind of build it up to a point where they really see promise in it and can kind of run with it from there. And then there'll probably be a lot of conversations with NGOs around who will pay for what and how do we pay for it. And I think the cost part is the part where I have the least experience, but I'm also really interested because I realize you want it to go somewhere. So trying to be in spaces where we can have those conversations. Paul Drain, who's our clinical partner, has this really amazing network of people. So I'm hopeful that we'll continue to learn more from there.
Thank you.
Yeah.
Well, I think we'll close it out here. I want to, before we say thank you so much, I just want to put a shout out for our next series of talks. We have 4 upcoming talks about mechanical engineering in space. So we'll have those back to back, except we don't have a seminar on election day. But if you guys come back for those, it'll be great. But let's go ahead and say thanks again to Ayo and do a round of applause.
Thank you, everybody. Thanks for showing up and for your questions and engagement. Thanks, Nate.